feel like a secret you send out that nobody knows. We're in a party, dreaming and tired. We're in a life that I regret.
Assalamualaikum warahmatullahi wabarakatuh. We are from group AS1205A. The title that we choose for our presentation is Chemical Profiling and Evaluation of Antioxidant and Antimicrobial Properties of Selected Commercial Essential Oils.
Hi guys, today I want to talk and discuss about the chemical profiling of essential oil. Firstly, I want to explain about what is essential oil. Essential oil is actually a concentrated hydrophobic liquid containing volatile chemical compound from the several parts of plant, such as flower, leaf, stem, seed, and fruit. And the complex chemical compound from the mixture of concentrated hydrophobic liquid can cause the essential oil to have a strong smell or odor. In this video, we want to discuss more about the chemical profiling of essential oil and the gas chromatograph mass spectrometry analytical method that been used in chemical profiling of essential oil. This method is actually a combination feather of analytic method, which is gas chromatograph and the mass spectrometric. And generally, this method or instruments is used to identify substance within test sample such as a drug and essential oil. And this method also very useful to many departments, especially for security and criminal forensic. In security, these instruments are used to detect the drug in fire investigation and in explosive investigation. And for the criminal forensic, these instruments are used to analyze nanoparticle and substance from human body that may link to the crime or case. Next, we want to discuss more about the mechanism and the principle of gas chromatograph mass spectrometric instruments. There are two major building blocks in this instrument, which is one block is for gas chromatograph and the other one is for mass spectrometric. And in gas chromatograph, the instrument are using a push and trap system, which is the inner gas such as helium gas, nitrogen gas will push the sample test that mix with the water to help them move along with the column in the ovens. The ovens with 300 degrees Celsius of temperature will help the separation process of the molecule and the separation process also can be caused of the difference of chemical properties and the difference of molecule in a mixture. After that, the molecule will retained by the column, and will come off from the column at the different time, or we call as a retention time. And during the retention time, the molecule will move out from the oven and be transferred to mass spectrometric block. And in the mass spectrometric block, the molecule has been captured and undergo ionization process, which is to break the molecule to ionized fragment. There are three ionization methods, for example, like electroionization, cool ionization, chemical ionization. After the ionization process, the ionized fragment will be detected by the electron multiply detector, and the spectra that being shown or that been detected will be recorded in the computer. Lastly, we want to talk about the objective of chemical profiling of essential oil. Basically, chemical profiling being used is to observe the properties of antibacterial, antimicrobial, antioxidants, and antiquorum in the plants. And we also went to observe the comparison of a chemical compound and the bioactivities of the essential oil in the same type of genus.
The first method is gas chromatography mass spectrometry analysis. The first step is essential oils were analyzed in an Agilent 7890. The identification of the compounds was performed using a time database that was tested against the mass database. An Agilent VF50 column with 30-meter line, 0.25 diameter, and 0.25 nanometer thickness was used. The assays were started at the temperature of 50 degrees Celsius for 5 minutes. Afterwards, the temperature was raised to 180 degrees Celsius at a rate of 2 degrees Celsius per minute, and final temperature was maintained for 30 minutes. The temperature of the injection port and transfer line was set at 230 degrees Celsius. The speed injection made of ratio 1:20 was adopted with helium as the carrier gas at a constant flow rate 1 ml per minute. The mass spectrometer was operated in the mode of electron ionization with an electron energy value of 10. The identification of the compounds was confirmed based on the retention indexes and their mass spectra. The next method is to determine antibacterial activity. The first experiment is bacterial strains and growth conditions. 10 bacterial strain was used for the antibacterial studies. Stock cultures of the bacterial strain were prepared and kept with 20% glycerol at 80 degrees Celsius. All the strains were subcultured in brain-heart infusion agar for 24 hours before antibacterial assays. The next method for determination of antibacterial activity is this diffusion assay. The first step for this diffusion assay is the antibacterial activity of the essential oil was evaluated by the diffusion assay. Inoculums were prepared by suspending bacteria in a sterilized saline solution to a cell suspension of 1 to 2 times 10 to the power of 8 CFU per milliliter. These with a diameter of 6 mm were saturated with 10 microliter of each essential oils. The MHA plates were inoculated, allowed to dry, and the discs prepared previously were placed over the agar. The inoculated plates were incubated at 37 degrees Celsius for 24 hours. After the incubation, all the plates were checked for inhibition zones. The diameters being measured in milliliter. Next is the evaluation of anti-quorum sensing activity. This diffusion test was used. For the first step, sterilize discs with 6 mm diameter saturated with 10 microliter of the essential oil were placed over the plates and incubated at 30 degrees Celsius for 24 hours. After the incubation period, the inhibition of the pigment production around the disc was measured. The lack of microbial growth indicates antimicrobial activity. The quorum sensing inhibition was then determined by the following equation.
Hi So today I will be presenting the results for this study. The results will be broken down into several different sections, including chemical profiling, antioxidant properties, as well as antibacterial and anti-quorum sensing properties. So based on the results obtained, more than 98% of the chemical volatile compound present in the essential oils were identified. So the chemical composition of the essential oils of the eucalyptus species are shown in table number 2. The major compound of E. citrodora is citronellal, which makes up about 78.15% of the oil. Citronellal is a monoterpenoid responsible for the strong lemon odor in the essential oil or in any essential oils from the citrus species. So this oil from E. citrodora is also known as lemon eucalypt. The major compound of E-SMIP oil is eucalyptol, which makes up about 81.71% of the oil. Eucalyptol is a monoterpene that is used for medicinal purposes, flavorings, and fragrances, and this compound exhibits mosquito repellency, anti-tumor properties, as well as anti-inflammatory activities. East Nigeriana oil has 29.18% of eucalyptol with 12.33% of bornyl formate, followed by 12.11% of isobornyl formate as its major constituents. The chemical composition of the essential oils of Lavandula species is recorded in Table 3. So the principal compound for the essential oils of L. angustifolia grown in Croatia is linalool at 66.83%, and for the same species but grown in France, linalool comes second at 37.31% after curcumin aldehyde, which is the major constituent of the oil. The same plant species grown in high altitude in France also presents a great amount of linalool at 23.49%, but with linalool formate at 41.72% being the most abundant compound. The major compounds for the other Lavandula species, which is the L. officinalis, are linalool at 47.86%, followed by linalool formate at 22.09% and limonene at 14.78%. Linalool is a naturally occurring floral and spicy terpene alcohol that can be found in many plant species, and it is used in multiple commercial applications. So this study indicated that the Lavandula species grown in Croatia has linalool as its major compound, while for French Lavandula species, it is found as the second compound present. Last but not least, the chemical composition of R. officinalis, C. citritus, and G. communis essential oils are presented in Table 4. The major compounds for R. officinalis is limonene at 46.89% and camphor at 11.94%. Next is the chemical profile of C. citritus, or more commonly known as lemongrass. The major compound of C. citritus oil is citronellal formate at 42.82%, followed by isobornyl formate at 33.52%. G. communis oil is majorly constituted by 1-propenyl 4-methylene cyclohexane at 32.29%, followed by D-cadenine at 11.28%. Cadenine is the chemical name given to a variety of isomeric hydrocarbons that occur in a wide variety of plants that is known to produce essential oil. Next, let's move on to the antioxidant properties of these essential oils. They were measured by DPPH, free radical scavenging assay, and beta-carotene leaching tests. They were summarized in Table 5. So the C. citritus oil has the best capacity to scavenge the DPPH free radicals, and it has an IC50 and AAI value significant enough for it to be easily classified as having a strong antioxidant activity or property. For the eucalyptus species, E. stageriana oil has the best antioxidant activity, followed by the oils of E. citrodora and E. SMIP. For the Lavandula species, the essential oils of L. angustifolia grown in France shows a great or strong antioxidant activity in comparison to the same species grown in Croatia, which shows moderate antioxidant activity. Now let's move on to the beta-carotene bleaching test. So beta-carotene bleaching test is an indirect measurement of the inhibition of lipid peroxidation by the essential oils. So the juniper species oil has the lowest number of IC50, and this indicates that it has the best capacity to inhibit lipid peroxidation chain reaction. Finally, we have the antibacterial and anti-quorum sensing properties. The diameters of the inhibition zones obtained for several gram-positive and gram-negative strains of bacteria tested in contact with the essential oils are presented in Table number 6.
Generally, the gram-negative strains are more resistant to the effect of the essential oils. Thus, it presents lower diameters of inhibition zones. It is also described that gram-negative bacteria should be less susceptible, which is associated with the characteristic of the outer membrane of these organisms that provide the bacterial surface with strong hydrophilicity acting as a robust permeability barrier. From the result, the essential oils of C. citratus stands out from the other oils since it presents great antibacterial activity. It can be demonstrated by the high inhibition zone diameters, which was verified through the bacterial strains tested. Among eucalyptus species, those with best antibacterial properties are E. citriodora and E. styrtheriana oils. However, for the lavandula species, all of the essential oils presented significant antibacterial properties. In addition, no relation was found between the antibacterial properties and the geographical origins of the essential oils of this species. The resistance and pathogenesis of the bacteria could be linked to the mechanisms of cell-cell communications. For example, quorum sensing. Quorum sensing is described as the response regulation system of genes expression in accordance with the variation of cell population density. These mechanisms allows gram-positive and gram-negative bacteria to regulate some physiological activities. The signal molecules that involved in quorum sensing is acylhomoserine, lactone, or known as AHL. From the result of this study, the essential oils of R. officinalis presented the greatest capacity to inhibit the violacin production by C. violacum. Thus, this indicates its great anti-quorum sensing activity. Next, among eucalyptus species essential oils, only E. citriodora has the capacity to inhibit the quorum sensing mechanisms in C. violacum. And lastly, for the lavandula species, only L. angustifolia, which grows in Croatia, is the only one that unable to block the quorum sensing process. AHL contains pigmented bacterium C. violacum, which normally produce the purple compound called violacin in response to the presence of N-hexanol homoserine lactone. The inhibition of this violacin production is linked to the inhibition of the quorum sensing. Therefore, the anti-quorum sensing activity of the essential oils also can be evaluated using the biomonitor strain C. violacum through this process.
Essential oils and a few of the isolated compounds are utilized in various products like cosmetics, household cleaning products, air fresheners, hygiene products, as well as in medicinal uses. Generally, within the genus Eucalyptus, the main compound present in the essential oil is eucalyptal, occurring in several concentrations. However, when watching the antioxidant properties, it had been noted that the oil with the highest amount of eucalyptal, which is the East Media oil, with a percentage of 81.71%, was the one with the weakest activity. These results indicate that the biological properties of the essential oils are determined by the interaction of their components. Besides, it was also noted that lavandula species essential oils are mainly constituted by linalool. But, however, specific species from different geographical origin also influence the chemical composition of the essential oil. The soil and climate are some critical factors that influence the chemical composition of the essential oils. The French lavandula species essential oil presented the simplest antioxidant activities in comparison to those from Croatia. The essential oil with high linalool content presents a lower antioxidant activity, in contrary to what's described for linalool and essential oil with this compound, which are mentioned as having relevant antioxidant activity. This fact shows once more the importance of synergistic or antagonistic effects which may occur among the numerous components of the essential oils. Concerning the antimicrobial properties of the essential oils, the oil of C. citruses resulted in greatest inhibition of the expansion of the bacterial strains under evaluation. It had been also possible to notice the presence of synergistic interactions among the compounds of the essential oil that influence the results obtained within the antibacterial test. An important issue to require into consideration when using essential oils in humans is that they can't be toxic. Moreover, it had been stated that the most compounds of R. officinalis essential oil should anti-mutagenic or anti-genotoxic potential against several carcinogens. Concerning linalool, a major compound found in several essential oil, it's also commented that it exhibited anti-mutagenic potential. Therefore, it is often inferred that generally and within the bioactive concentrations, essential oils are safe to humans.
In conclusion, eucalyptol is the principal compounds of the essential oils of eucalyptus species, and linalool is the principal compounds of the essential oils of lavandula species. The geographical origins of the plant species are determinant factors in the essential oils compositions and in the corresponding biological activities. The essential oils of Cymbopogon citratus presented remarkable antioxidant and antibacterial activities, which comes together with its pleasant odor that makes this oil a promising natural agent to be used in both of the pharmaceutical or food industry. Okay, this is the resource that we use to complete this presentation. That's all from us. Thank you.